All right, now we're going to go over Chapter 6. This is going to be an introduction to viruses, varioids, and prions. This is an interesting chapter. So to talk about viruses, first we need to go back a little ways and talk about Koch's postulates. So remember that Robert Koch came up with a series of logical steps to identify microbes causing disease. And he identified several pathogens, including tuberculosis, cholera, and anthrax. Now, all of these pathogens that he identified are bacteria. He was not able to identify any viruses using the postulates because the postulates require that you culture the microbe, grow it, and viruses are very difficult to culture. So for many years, the cause of viral infections was unknown. Louis Pasteur came along and hypothesized correctly that rabies was actually caused by something smaller than bacteria. He thought it was a living thing. We'll talk about that in a minute, why viruses aren't exactly living. Pasteur proposed the term virus, which is Latin for poison. Then you got these guys coming along, Loeffler and Frosch. They described a filterable agent that was the cause of certain diseases. So way back when, they would filter these fluids through porcelain filters that are designed to trap very small organisms like bacteria. And when they did this, they would notice that sometimes the filtrate remained infectious. This is because there was something smaller than bacteria slipping through those holes in the filter. And so this proved that an infection could be caused by a fluid containing agents smaller than bacteria. Okay, so where did viruses come from? Well, the short answer is we don't know. There is no universal agreement on the origin of viruses. Now, most researchers think that viruses evolved after cellular organisms, but we don't know. They could have been first. What we do know is that they are more numerous than all the cells on the earth. There's about 10 viral particles for every bacterium. Seawater can contain up to 10 million viruses per milliliter, and believe it or not, you have about 140,000 different viral species living just in your intestines. Many of these are bacteriophages, and we'll talk about those in a little bit. What bacteriophages are is they are viruses that infect just bacteria. So the thought is that these viral species living in your intestine help to keep the bacterial populations in check by infecting different populations, bringing their numbers down, and preventing one bacterial species from overgrowing. Viruses are also known to have played some sort of role in the evolution of bacteria, archaea, and eukarya. And we know this because we can look at our genomes and we can find viral DNA. In fact, about 8% of the human genome is viral in origin. Okay, so viruses have been around for a long time and they've had long-lasting effects on the organisms that they infect. So let's talk just a little bit about the properties of viruses. So viruses are obligate intracellular parasites. This means that they cannot do anything on their own. They have to get inside of a cell and hijack the cell's machinery in order to replicate and produce more viral particles. They do not have machinery of their own. Now, they're parasites of bacteria, protists, which remember are going to be your algae and your protozoa, fungi, plants, and animals. So viruses can infect every living thing. Viruses are also tiny. They are ultra microscopic in size, so you need something stronger than a light microscope to visualize them. They are not cellular in nature, so they don't have a cell membrane, they don't have organelles, they don't have a cytoplasm, okay? They're very compact, very economical. And we'll talk about that here in just a minute. On their own, viruses do not fulfill the characteristics of life. So we do not speak of viruses as being alive or dead because they are not living creatures. Instead, we talk about viruses as being active, that is able to infect, versus inactive or unable to infect. Okay, so outside of the host cell, Viruses can do nothing, okay? So they're going to be inactive until they get inside of a host cell. Then they're going to be active. They can replicate. The basic structure of a virus is going to be a nucleic acid core, so either DNA or RNA, wrapped in a protein shell, which we call a capsid, okay? Very, very simple structure. The nucleic acid core of the virus is going to be either DNA or RNA. It is never both. The nucleic acids can be double-stranded DNA, single-stranded DNA, single-stranded RNA, or double-stranded RNA, okay? So the nucleic acid can take different forms depending on the virus. Okay, and then viruses have molecules on their surface that are going to be highly specific for attachment to a particular host cell. For example, respiratory viruses in general are going to infect your respiratory epithelium. They're not going to infect other cells of your body like your liver cells, okay? But there are viruses, for example, hepatitis viruses that have a specificity for your liver and not for your respiratory epithelium or any other tissue in your body, okay? So it just depends on the virus and what it is specialized to attach to. How do viruses work? Well, they're going to take control of the host cell, and they're basically going to use that host cell's machinery to create more nucleic acid and more protein to coat that nucleic acid, right? And then they're going to release that. That's what a virus is. Nucleic acid with a protein coat. They lack enzymes for most metabolic processes. So this is in general, but there are some viruses, notable viruses that do have enzymes. For example, HIV has two enzymes here, reverse transcriptase and integrase. Now, HIV is an RNA virus. This means it makes RNA. But what reverse transcriptase does is it takes that RNA and it turns it into DNA. And then integrase takes that DNA and it inserts it into the host cell's genome. So a patient who is infected with HIV will have HIV DNA in their genome, in their cells. And then I've already said multiple times, viruses are going to lack machinery. They lack machinery for synthesizing proteins. They lack machinery for synthesizing their nucleic acids, okay? So they need the host cell's machinery. All right, general size of viruses. Just know they are ultra microscopic. They're very, very small, and you're going to need an electron microscope to see these guys. So you can see the comparison here. We have E. coli. It's about 2 micrometers long. You have Streptococcus. It's about 1 micrometer wide. Then you get down into the Rickettsias. These guys are pretty small bacteria. And then we're into the ultra microscopic range. So here's mimivirus. You can actually see these with the microscope. These are very large viruses. And they can even stain with the Gram stain, believe it or not. You've got pox virus, herpes simplex, rabies. We're getting smaller, 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 smaller. Then you can see here is a hemoglobin molecule. This is what carries oxygen in your red blood cells. And next to it, we have the poliomyelitis virus. And this is a very small virus. So you can see some variation in the size of viruses, but by and large, they're going to be ultra microscopic. You cannot see these with a standard microscope. Okay, this here is an electron microscope image of a mimivirus. Okay, this is a very, very large virus. These arrows are pointing to actually smaller viruses surrounding the mimivirus. So you can get an idea of just how big this virus is compared to these guys. It's so big, in fact, that mimiviruses can stain Gram positive. The Gram stain gets caught in the capsid of the virus. And you can see that here is amoebas that are infected with mimivirus. And if you look at the amoeba, you can see these fine little purple granules. Those granules are actually viral particles, okay, that have been stained with the Gram stain. Right, a little bit about viral structure. We said the viruses bear no resemblance to cells, okay? They don't have protein synthesizing machinery. They don't have a nucleus. They don't have organelles. They don't have a cell membrane. They don't have cytoplasm. They don't have any metabolic capabilities. They contain only the parts that they need to invade and control a cell. The virus particle itself is going to have a covering and then a core. The covering will always include a capsid. Capsids are proteins, okay? Protein shells. Sometimes some viruses will have an envelope. We'll talk about that in a minute. Then beyond the covering, there's going to be a central core. And this is going to be made up of a nucleic acid molecule, either DNA or RNA. Never both. That's important to know. In general, but also for your test. Sometimes the central core will have enzymes and matrix proteins, but remember, this is not found in all viruses. All right, so I mentioned the capsid. This is a protein coat that goes over that nucleic acid, okay? So it goes over that DNA or that RNA. The capsid, along with the nucleic acid core together, we call that the nucleocapsid. Some viruses have an external covering called an envelope. We'll talk about that in a second. But the ones that don't have an envelope are called naked viruses. Okay, so this would be an example of a naked virus here. Each capsid is made up of identical protein subunits called capsomeres. So if you look here, this kind of black squiggly stuff in the middle is the nucleic acid. This blue polygonal shape here is the capsid. And you can see that the capsid is made up of these individual units that are repeating, and these individual units are called capsomeres. All right, so this is showing us a naked virus. This outer shell here is the capsid. Then you can see the nucleic acid in the middle. This whole unit you would call together would be called the nucleocapsid, okay? Next, we have an enveloped virus. You can see the nucleocapsid inside, right? So the capsid and the nucleic acid core. But on the outside is this membrane, and we call it an envelope. And where this membrane derives from is actually from the host cell itself. And you'll see embedded in this envelope are these spike proteins. And these spike proteins are going to be very important for attachment of the virus to its given host cell. So what kind of shapes can viruses take on? Well, we're going to talk about three basic shapes. Helical, which is going to be a helix of capsomers that forms kind of a cylindrical nucleocapsid. Icosahedral, this is going to be a shape with 20 sides and 12 corners. And then complex. Complex viruses have very unique shapes, such as bacteriophages, and I'll show you that here in just a sec. So here you can see an example of a helical virus. You can see the RNA is kind of wound into a spring, and the capsid kind of encircles it, creating a cylinder. Then you've got polyhedral viruses. You've got the nucleic acid inside, and then usually an icosahedral shape, so with 20 sides, but they can be other shapes as well. Spherical viruses, don't worry too much about this. All this is trying to show you is that a virus can have an envelope, and when it does, it tends to look spherical. And then here, I want you to know about complex viruses like bacteriophages. So this is a bacteriophage. You can see it kind of resembles like a lunar lander. It has a head, it has a tail, it has tail fibers. And what this thing does is it lands on a bacterium and it injects its nucleic acid into that bacterium, kind of like a syringe. Very interesting viruses. All right, enveloped viruses. You are going to see these mostly in animal viruses. So a lot of the viruses we'll be talking about in this course are going to have an envelope. Where does the virus get this envelope? Well, when it leaves the host cell, it takes a little bit of the host cell's cell membrane with it, and it creates an envelope around itself. Now, it also embeds proteins into this envelope. And these proteins are called spikes or spike proteins. You can see them here. And what they do is they are important for attachment to the host cell. So when this virus is released and it goes and finds another cell that it wants to infect, it's going to use these proteins, the spikes, to attach to that cell. So what is the purpose of a capsid and or an envelope? Well, these structures protect the nucleic acid when the virus is outside of the host cell. And they're also going to help the virus bind to the cell surface, like we said with the spike proteins, and assist in penetration of the viral DNA or the RNA into that cell. Okay, a little bit more on complex viruses. We've got a couple examples here. We have pox viruses. These guys lack a capsid and instead are going to be covered by a double membrane. And then we've already mentioned bacteriophages and how interesting their shapes are. So here is a pox virus with its double membrane. Then over here is a bacteriophage. You can see the capsid head here, which holds the nucleic acid core, and it's through the sheath and out this way that the nucleic acid is going to be transferred into the cell. I don't want you to worry about memorizing these guys, okay? This is just general information to give you a sense of how diverse viruses are. There are many exceptions to every rule when it comes to viruses because they are so unique. Here is an electron micrograph of a bacteriophage. Okay, questions. So, what is the main function of the nucleocapsid in a virus? Well, remember we said that the nucleic acid needs to be protected when it's outside of the cell. And what's going to do that? The capsid and or the envelope, if there's an envelope. Both of those structures protect the nucleic acid. So the answer here would be B, to protect the viral genome. Okay. Which of the following is true about viral envelopes? The viral envelope is always made of protein. The viral envelope is derived from the host cell's plasma membrane. The envelope is not essential for viral entry into the host cell, or the envelope is composed only of viral proteins, not host cell materials. Well, remember that the envelope, we see this in what? In primarily animal viruses. And where is the envelope obtained? Well, it's obtained from the animal cell. When the virus goes to leave it, it takes a little bit of its plasma membrane with it. So the answer here would be B, the viral envelope is derived from the host cell's plasma membrane. Which of the following is not a characteristic of a nucleocapsid? A, it consists of the viral genome and surrounding protein coat. No, that's true. B, it protects the viral genome from environmental damage. That's true. C, it is present in all viruses, both enveloped and non-enveloped. This is also true, okay? There is always going to be a capsid. There's always going to be nucleic acid. Or D, it is made of lipids and proteins. Well, D is incorrect. Remember that the nucleocapsid, the capsid is made of proteins, and then it surrounds nucleic acids, okay? So nucleocapsids are made of proteins and nucleic acids, not lipids. All right, so again, that viral genome we said is going to be either DNA or RNA, never both. Please remember that. The virus is going to carry just the genes necessary to invade the cell and redirect that cell's activities to make more viruses. So the number of genes for each virus is going to vary, but in general, it's going to be, they're going to have many fewer genes than other organisms. All right, DNA viruses. Usually the DNA in DNA viruses is double-stranded, but there are some viruses that have single-stranded DNA. I know that's weird. Viruses are weird. RNA viruses. Usually these are single-stranded, but because viruses are weird, sometimes the RNA is double-stranded. Additionally, the RNA can be broken up into separate pieces, and we call that segmented viruses, okay? Now, RNA is interesting in that it can be written in a couple of different ways. It can be written kind of in a forward way where it's ready to be read by the cell's machinery, or it can be written in a backwards kind of way where it needs to be converted to its forward way before it can be read by the cell's machinery. When it's written in its forward way, when it's ready for immediate translation by the cell, that's called a positive sense RNA virus, okay? If instead the virus's RNA is written backwards and it needs to be converted to a positive sense RNA first, then we call it a negative sense RNA. Okay, so here we have an example of a positive sense single-stranded RNA virus. It's entering the cell, and now its viral RNA is inside the cell. Now, because this is positive sense, it is ready to be translated. So we can take this strand of RNA, read it, and make proteins and all the viral mechanisms out of it. On the other hand, here's a negative sense single-stranded RNA virus. It comes into the cell. You can notice it's written backwards. So see how this end has a 5, this end has a 3? Well, it's backwards here. This end has a 3 and this end has a 5. So, and those are just indicators on which way to read the molecule, okay? So negative sense is backwards. So what we have to do is we have to make a complementary strand, okay? So here we're copying it and we're making another RNA strand that's complementary to the negative sense. So this is going to be written in the forward direction, the correct way. Now it's ready for translation. All right, other substances that viruses can bring along. Well, we mentioned some viruses bring their own enzymes. For example, polymerases. These are enzymes that synthesize DNA or RNA. Some viruses can hijack the host cell's polymerases, but the host cell has DNA in it, so its polymerase is always going to make DNA. If it's an RNA virus, it needs to bring along its own polymerase, okay, to make RNA. Replicases copy RNA, and then we talked a little bit about reverse transcriptase, and remember this synthesizes DNA from RNA, and we see this in the HIV virus. HIV can make DNA out of its RNA, and then it uses an enzyme called integrase to take that DNA and insert it into the host cell's genome. Okay, so how are viruses classified? Well, just know that it's complicated, okay? They use structure, chemical composition, and the genetics of the virus to classify them. And we put them into families and genera, okay? So families are going to end with Viridae, and then the genus is going to end in virus. Now, a viral species is a distinct viral type that shares a collection of properties such as the host range, so what cells it can infect, what animals it can infect, or organisms, I should say. Its pathogenicity, so how likely is it to infect you? And its genes. There's a bunch of different characteristics for placement in a virus family. Don't worry too much about this. Just know that it's complicated. And I want you to know that names are often acronyms that blend several characteristics together, such as the picornaviruses. This name comes from pico, meaning small, RNA, which means this is what? An RNA virus. Okay, so this just means small RNA viruses. Okay, this is just a chart going over some of the viruses that we'll be talking about, the DNA viruses we'll be talking about later in this course. You can see how they're named here, herpes viridae. So which viruses do you think this would include? Well, the herpes simplex 1, herpes simplex 2, varicella zoster, and even human cytomegalovirus is in this family. Hepadinaviridae, as you might guess, this has to do with hepatitis. So hepa being hepatitis, DNA being a DNA virus, okay? All right, here's a chart showing the RNA viruses that we'll be talking about over the course of this class. Lots of RNA viruses to cover, okay? And these are some of the most interesting viruses you'll find. Coronavirus is an RNA virus. All right, the viral life cycle. I want you to know this. It's also called the multiplication cycle. Okay, so the first step is adsorption. This is when the virus binds to the host cell. Next is penetration. This is just the virus entering the host cell. What has to happen next is you have to take that capsid off so that the viral nucleic acid is released and it can be copied. So that step is called uncoating. You're uncoating the capsid, okay? Next is synthesis. This is where the viral components are going to be produced. Assembly, the viral components are put together. And then finally, release, where the assembled viruses that are ready to be released are released either by budding. This is exocytosis. This is where they come out of the cell and take some of that cell membrane with them. Or through cell lysis. This is where they burst out of the cell, break open the cell, and kill the cell. And here's an image just showing the steps of the viral multiplication cycle. So we have the first step here, adsorption. Look, we're looking at a enveloped virus, right? So the envelope, the spike proteins on the envelope are going to bind to receptors on the cell membrane. When they do, that's called adsorption. Next, the virus is going to penetrate. So it's engulfed by the cell. The cell is taking this virus in unknowingly to its own detriment. Next, the virus is going to uncoat. It opens up its envelope and capsid, lets out the nucleic acid. Next is synthesis. So the cell is going to synthesize the new components of the virus under the control of the viral genome. Then assembly. All of these components are going to be assembled together to create new viruses. And then finally, release. So you can see here we have a capsid and the nucleic acid core, but there's no envelope yet. The virus obtains its envelope when it buds off or is exocytosed from the cell. Okay, so a little bit more about adsorption. This is going to happen when the virus collides with a susceptible host cell. And the receptors on that host cell are going to bind to the proteins on the virus. Now, the spectrum of cells a virus can infect is called the host range. This can also be referred to as tropism. Different viruses have different cells that they like to infect. For example, hepatitis B infects human liver cells. Poliovirus infects primate intestinal and nerve cells. Okay, so it's not just limited to humans. And then rabies infects mammals, particularly neurons, muscle cells, and the salivary glands of mammals. Here's an image up close showing adsorption. So you can see the receptors on the host cell membrane. And you can see this virus particle kind of colliding and its spike proteins connecting with these receptors. That's the first step of the viral multiplication cycle. Next, you have the steps of penetration and uncoating. Most viruses are going to penetrate through one of two ways. Either fusion, this is going to be only with enveloped viruses because they have that membrane around them, the envelope. And this is when the viral envelope fuses directly with that host membrane, and then the nucleocapsid is kind of dumped into the cell. The other way is going to be endocytosis. And this can happen with either enveloped or naked viruses. So the entire virus is engulfed by the cell. And then the cell takes it in as a vacuole. And then once inside, the virus is going to uncoat itself. And then the nucleic acids are released. All right, so an example of fusion versus endocytosis. So here we have attachments, right? Those spike proteins are connecting with the receptors on the host cell. Next, though, here we have membrane fusion, and you can see this enveloped virus, the envelope itself fuses with the cell membrane, and then the capsid, the nucleocapsid is just kind of dumped inside. Compare this to entry by endocytosis. You can see the cell is engulfing the virus, the whole virus, including the envelope. So now you have a vacuole containing an enveloped virus, and the nucleocapsid exits that vacuole and is uncoated from there. Okay, replication. Where does this happen? Well, it depends on whether the virus is a DNA or an RNA virus. DNA viruses are going to get into the nucleus, and they're going to be replicated and assembled there. This is because DNA viruses use the host cell's DNA polymerase to synthesize its DNA, and that DNA polymerase is where the DNA is in a host cell. It's in the nucleus. On the other hand, RNA viruses, they don't need that DNA polymerase to replicate. They need RNA polymerase to replicate, and many of them bring their RNA polymerases with them. So they can replicate in the cytoplasm. They don't have to go through the step of getting into the nucleus. We already mentioned positive sense RNA. These guys are ready to go. Negative sense RNA has to first be made into positive sense RNA, and then it can be replicated. Assembly. So the mature virus particles are going to be constructed from this growing pool of parts. Generally, the capsid is the first thing that's laid down, and electron micrographs taken during assembly are going to show masses of viruses, and I'll show you right here. We have the presence of herpes simplex virions inside of the nucleus. So here is the cell membrane out here. Here is the nuclear envelope, this line here, and you can see inside of the nucleus are all of these virions, these viral particles ready to go. They are being assembled. Now remember, herpes simplex is a DNA virus, so it's going to be replicated and assembled in the nucleus. If this were an RNA virus, we would see the virions out here in the cytoplasm. Release, we already talked about. It's going to be one of two ways. They're either going to bud or be exocytosed, right, if it's enveloped. In this way, the viruses are released and the cell is not immediately destroyed. The other way that viruses can be released is they can break open the cell. This is lysis or rupturing. And this involves viruses breaking open the cell for release, and the cell ends up dying. All right, again, you can see release here. You've got your virus here that has made spike proteins, and these proteins are getting embedded into the cell membrane. And then the virus is going to attach to that portion of the cell membrane and bud off from that area and take that membrane with it and all of the viral proteins that it just embedded into that membrane. And you can see the process here, this exocytosis or this budding off. Viruses commonly contain both DNA and RNA. False. You got to know that for the test, okay? Viruses never contain both. They contain either or, okay? All right, so we said you can't see viruses under a microscope, but you can see the effects of viruses under a microscope. These are called cytopathic effects. This is virus-induced damage to the cell that alters its microscopic appearance. You can see gross changes in shape, size, and orientation of the cell if the cells have orientation. You can see development of intracellular changes. We see things called inclusion bodies, which are masses of viruses or damaged cell organelles. And then you frequently see syncytia. This is the fusion of multiple damaged host cells, and they fuse together to form a single large cell containing multiple nuclei. And we call these large cells giant cells. All right, here's an example of RSV. So respiratory syncytial virus, or RSV, is so called because it creates syncytia. What happens is this virus has fusion proteins on its envelope, and those fusion proteins get placed on the cell membrane when the viruses enter. And those fusion proteins can cause the cell to fuse with other damaged viral infected cells, okay? And they fuse together and they form one giant cell. And you can see one here, huge cell with multiple nuclei. Here's another example. Here's a giant cell here with multiple nuclei. Here's a giant cell here with multiple nuclei, okay? So that's all that the syncytial virus does, is it creates syncytia. The problem with this is that when these host cells fuse together, they lose their ability to function like they normally would. Okay, so syncytia can be very problematic. All right, and then here's some examples of the cytopathic changes that you can see. One example I have down here is the rabies virus. So you're not going to see a change in the cell shape, but you are going to see these inclusions, these inclusion bodies in the cytoplasm. They're called Negri bodies. You can see here, this is a neuron, okay? So here's the cell body of the neuron. And inside of it, here's the nucleus. Inside of it are these bodies here, Negri bodies. And this is just an example of some of the effects that viruses have on the cells. So persistent infections, also called chronic infections. This happens when the cell harbors the virus and is not immediately lysed. These can last for weeks or for the lifetime of the host. Examples, hepatitis C and HIV, those are two big examples right there. Many of these persistent infections can periodically reactivate under the influence of different stimuli, so physiologic stress, psychological stress, other illnesses, things like that, can sometimes reactivate these viruses. And when that occurs, this is known as the chronic latent state. And we have two examples here. We have the herpes simplex virus. Herpes simplex 1 is responsible for cold sores. Herpes simplex 2 is responsible for genital herpes. And as you probably know, if you are infected with herpes, it tends to reactivate. It tends to come back. The herpes zoster virus. This is the virus responsible for chickenpox in the initial infection, but years and years later, it can reactivate and come back as shingles. All right, let's talk a little bit about transformation. So some viruses can enter the cell and permanently alter its genetic material. We've talked about that. But some of them can do this in a way that it results in cancer. Now, the viruses that can permanently alter genetic material and cause cancer are called oncogenic viruses. The effect they have on the cell is called transformation. So transformed cells are going to grow faster. They're going to have alterations in their chromosomes, right, in the DNA material. And they're going to have the ability to divide indefinitely. And all of this is going to result in tumors. Oncogenic viruses are called oncoviruses. And here's a couple examples. We have the human papillomavirus. This can cause cervical cancer. And we have the Epstein-Barr virus, or the common cause of mononucleosis, which rarely can result in Burkitt's lymphoma. Okay, so I have an example here of normal cells on the left. And these are squamous cells, so they're real thin, they're flat, kind of waifish. You can kind of see this cell here is kind of folding back over itself. Okay, and then over here are transformed cells. Same kind of cell, squamous cell, but you can tell they're larger. They have larger nuclei. They have more genetic material. They have darker nuclei. It stains darker because there's more DNA there to absorb the stain. And then you can see they are multinucleated. The overall big picture here that you're seeing is transformed cells are going to lose the ability to regulate the cell cycle. All right, so they're going to grow quickly. they're going to divide over and over and over. And sometimes they're going to have aberrations in their DNA and even have multiple nuclei because their ability to control the cell cycle is impaired. Bacteriophages, I mentioned before, these are going to be viruses that infect bacteria. We also call them phages. The most widely studied of these are going to be the kind that infect E. coli, and their multiplication is going to go through similar stages as the animal viruses. However, only the nucleic acid enters the cytoplasm, so the uncoating step is unnecessary in the bacteriophage multiplication cycle. Bacteriophages are released as a result of cell lysis, and this lysis is going to be induced by viral enzymes and the accumulation of viruses, and this is all called the lytic cycle. All right, steps in phage replication. You're going to notice this is very similar to the viral multiplication life cycle we've already talked about. You've got the step of adsorption, where the virus is going to bind to the host cell. Penetration, where the virus or the viral genome enters the host cell. But you'll see there is no uncoating step. You go straight into replication, where the viral components are produced. Then they are assembled. Then here's a new step, they mature. We didn't talk about this when we were talking about animal viruses, but in bacteriophages, there's the maturation step. Next, you have lysis and release, right? So the lytic cycle is going to be where the viruses break out of the cell and kill the cell as they are released. Occasionally, however, the virus is going to enter a state of lysogeny. And what this is the virus incorporates its DNA into the host's genome and just hangs out. It doesn't hurt the host, and the host reproduces, divides, and produces daughter cells. And each one of those daughter cells is going to have that viral DNA in its genome. And this continues until a point when the virus is induced somehow. And it's basically reactivated and then it enters the lytic cycle. Now for the test, there will be a question over viral multiplication, and you will need to name these steps in order, okay? You need to know adsorption, you need to know penetration, you need to know replication, you need to know assembly, maturation, and finally you need to know release. The virus is released somehow, okay? So a little bit more on lysogeny. Not all bacteriophages are going to go on to complete the lytic cycle. Some phages, called temperate phages, are going to undergo adsorption and penetration, but they don't replicate. Instead, the viral genome gets inserted into the bacterial genome and is then referred to as a prophage. It's inactive at this point, and the cell is not lysed. The prophage hangs out in the bacterial genome, and it's copied during cell division. So this means that all of the progeny of the host cell are going to have the prophage in their genome. This is lysogeny. At some point, induction can occur, and induction is going to result in activation of the prophage. And then the virus is going to re-enter the viral multiplication cycle, okay, and undergo viral replication and then lyse the cell. All right, so let's go through the multiplication cycle of bacteriophages. So here we have our E. coli host. Here is its genome. And here is the bacteriophage, and it has landed on the E. coli. It is adsorbing to the E. coli bacterium. Next, the bacteriophage is going to penetrate the E. coli cell, and basically it acts like a syringe and kind of squeezes the genome through the cell membrane and into the cytoplasm. This is penetration. Next, the phage components are going to be duplicated. This is going to result in replication of the virus genetic material. Next, you're going to have assembly. Then you're going to have maturation. And then when these virus particles are ready to leave, they're going to burst out of the cell, lysing it, breaking it open, and the cell is going to die. However, some phages can enter the lysogenic state. So let's start over. Here we have our E. coli host. Here's the bacteriophage. The bacteriophage is infecting the host. But now in lysogeny, it stops here. You have adsorption and penetration. Then you switch over to the lysogenic state. And what happens, you can see here, is the bacterial genome. Here is the viral genome. And the DNA is going to split open and incorporate that viral genome into its genome. So now we have a chromosome that looks like this. This cell in the lysogenic state is going to undergo cell division, and all of the little E. coli bacteria that result from it are going to have this genome with the virus, the viral genes in it, okay? At some point, induction is going to occur. And induction is going to basically wake up this phage and bring it back in to the bacteriophage multiplication cycle, so that it's going to start replicating, it's going to undergo assembly, it's going to undergo maturation, and eventually release. Now, lysogeny is going to result in spreading of the virus without actually killing the host cells. Now, what's kind of interesting about this is that some bacteriophages have genes that can produce toxins or enzymes that cause problems in us. And when they do, this is called lysogenic conversion. Let's go through an example. Corynebacterium diphtheriae. This is the bacteria that causes diphtheria, but only certain strains of it cause diphtheria. Only the strains that have been infected with a bacteriophage that contains the genes to produce diphtheria toxin. So in order to get diphtheria, not only do you have to be infected with Corynebacterium diphtheriae bacteria, but they have to be bacteria that have also been infected by a very specific bacteriophage carrying the genes for the diphtheria toxin. Now, if you're unlucky enough to get infected by bacteria that have those phage genes in them, you can develop diphtheria. And there's a lot of other examples. For example, Vibrio cholerae has a toxin it produces. Clostridium botulinum has a toxin it produces. E. coli and Shigella each have a toxin that is identical to one another, so it is spread by bacteriophages. So you see this quite frequently. All right, here's just a closer look at the penetration step. You can see the bacteriophage here. Here's the head. This normally is where the nucleic acid is. And here you can see it's penetrating. It's in the penetration step. So it's squirting its nucleic acid out of the bacteriophage and into the cell itself. This I just thought was a cool picture to show you. It's a bacterium. You can see the cell membrane here, and you can see all of these bacteriophages that are lined up on it, as well as these bacteriophages that are bursting out from it. This is a good chart to look over. This is just giving you a comparison between bacteriophage and animal virus multiplication cycles. One thing I just want to draw your attention to here is under viral persistence. In bacteriophages, we see this expressed as lysogeny, right? The lysogenic cycle. However, in animal viruses, viral persistence is going to look different. It's going to be either latency, so latent viruses like herpes, chronic infection like hepatitis C, or cancer is another way that the virus persists in causing cancer. All right, which of the following is a step found in animal virus multiplication, but not in bacteriophage multiplication? Well, the answer here is going to be C, uncoating. Remember that the bacteriophage injects its viral genome directly into the cell. There's no need to uncode it from the capsid. Okay, animal viruses are the ones that undergo uncoding. Cultivating viruses, aka growing viruses, so to speak. What is the purpose of cultivating viruses? Well, you want to isolate and identify viruses in clinical specimens. So if you have a patient you suspect has a viral infection, you want to be able to isolate and identify that virus. We prepare viruses for vaccines. And of course, detailed research on viral structure, its multiplication cycle, its genetics, and so on. So these are all reasons why we would need to grow or cultivate viruses. Now, I mentioned before that Robert Koch's postulates could not prove viruses. And one of the reasons is because his steps require you to cultivate the microbe in a lab somehow. So to grow the microbe. And viruses just don't grow like that. Viruses you have to grow inside of cells somehow, right? That's the only way that they can replicate themselves, is if they are inside of a cell. So there's two main ways to cultivate viruses in cells. There's the in vitro method. Okay, this means in glass. And this is done in the lab. This is where cultured cells support the viral replication, and you can observe this basically sheet of cells that you have infected with virus. So that's how you do it in the lab. In vivo, or in life, you can use living animals to cultivate virus. So one way we do it is with bird embryos. The nice thing about this is that they are a self-supporting unit with inside the eggshell, and it's a sterile environment in there with its own nourishment. So you can infect a bird embryo and grow those viruses in its cells without really having to ever feed it or do anything special to the embryo. You can also inoculate a living animal. So what they do here is they take an animal, they inject it with virus, and then they watch the animal for signs of infection and study it that way. Okay, so why do we care about viruses? Well, they are medically very important. They're the most common cause of acute infections. There's several billion viral infections per year. And some viruses are going to have a very high mortality rate if left untreated. For example, rabies, fatality rate of rabies untreated is almost 100%. HIV has a very high mortality rate if left untreated, higher than 90%. Some respiratory viruses also have high mortality rates, and some hemorrhagic diseases, hemorrhagic fevers as well. Then there's a possible connection of viruses to chronic diseases that we don't know the cause of yet. For example, there's a lot of evidence to suggest that several autoimmune disorders might be triggered by viral infection, such as type 1 diabetes, multiple sclerosis, or Crohn's disease. Then there's also some evidence linking neurodegenerative disorders to viral infection as well. So examples there would be Alzheimer's disease and Parkinson's disease. Viruses also participate in the Earth's ecosystem. Even though we don't consider them living organisms, their effects on living organisms are large enough to help shape the Earth's ecosystem. Detecting and treating viral infections. This is going to be pretty difficult, more difficult than detecting and treating a bacterial infection. One of the things that providers need to do when they are assessing a patient and considering whether it's bacterial in origin or viral in origin, they need to consider the overall clinical picture. So don't focus too much on what one lab result says. An appropriate sample should be taken to screen for either parts of the virus, screen for an immune response to the virus, such as antibodies, or to infect a cell culture. So you could take a sample, use that sample to infect a whole bunch of cells, then look at those cells under a microscope for cytopathic effects. You don't want to go giving every patient with a potential viral infection antiviral drugs, because they are some heavy hitters and they can cause serious side effects. So most viral infections are probably going to go untreated because we don't have very good antiviral drugs for most viral infections. And antibiotics, as the name suggests, work on living things, right? Antibio. So a virus doesn't have the structures that that antibiotics are targeting. So antibiotics don't work on viral infections at all. Okay, next we're going to talk just a little bit about prions. Prions are infectious particles. Basically what they are is just a misfolded protein. There's no nucleic acid. There's really no structure to it, just a little protein that's been misfolded. These tiny proteins are resistant to usual sterilization techniques, so they are very hard to get rid of. And they can cause fatal neurodegenerative diseases called transmissible spongiform encephalopathies. The greatest example of this, everybody knows, is mad cow disease or bovine spongiform encephalopathy. But you also see scrapie in sheep and goats, wasting disease in elk, and when prion disease affects us, we can see Creutzfeldt-Jakob syndrome. All right, so what are we looking at here? Well, this is a nerve cell. This is the body of the nerve cell. And here we see prion proteins invading the cell, okay? So these are little misfolded proteins that come into the cell, and they interact with normal proteins. And when they interact with normal proteins, they cause the normal proteins to misfold as well. And these misfolded proteins, these prions, form aggregates, and these aggregates cause dysfunction in the cell. Okay, other non-cellular infectious agents, there are viruses called satellite viruses. And what these viruses, what makes these viruses so special is they depend on other viruses for replication. So it's kind of funny, you know, a normal virus depends on a cell for replication, while a satellite virus depends on a virus for replication. Probably the most obvious example here is going to be hepatitis D, also called the delta agent. And this is just a strand of RNA that is expressed only when the cell is co-infected with hepatitis B. Then you have viroids. These are just short pieces of RNA, and so far have only been identified in plants. Okay, question. Exposure to nucleases, those are enzymes that degrade or break down DNA and RNA, would damage all of the following except, okay, so we're looking for something that does not have DNA or RNA in it. So animal viruses are always either DNA or RNA. Bacteriophages have nucleic acids. Satellite viruses have nucleic acids. And viroids, we just learned, is a short strand of RNA. The only one in here that does not have nucleic acid are prions. Prions are misfolded proteins, okay? So something that degrades nucleic acids is not going to affect a protein.